And, okay. So we were starting to talk about the components of a cell last week, and in doing this, I have a video that we're going to watch to start, just to make it a little fun. So I have to switch my screens. One sec. Okay, so we're going to start with this song about the cell.
I went into a cell to get out of the rain. And there was the gatekeeper, the cell membrane. I went into a cell, and what did I see? The mitochondria, it's the energy factory. I went into a cell and said, who drives this bus? And found myself talking to the Boston nucleus. Went into a cell to recover from a spasm and found myself swimming in some clear cytoplasm. I went into the nucleus to ask how to get home and got genetic info stored in a chromosome. Went into a cell and stretching oh so far was a thin and wavy network. It's called the ER. I went into a cell trying not to be perplexed by the packaging and sorting in the Golgi complex. I went into a cell and said, who makes proteins here? And somebody responded, it's the ribosome, my dear. I went into a cell and was feeling pretty fine till a lysosome engulfed me and dissolved me in enzymes. I went into a cell and was feeling pretty nimble till a centriole lassoed me, tying me up in a spindle. I went into a plant cell to see how trees get so tall, and all around the outside was a rigid cell wall. I went into a plant cell. Why is it so green? I asked. Cause I make food from sunlight, said a green chloroplast. I went into a plant cell to see how plant cells store food. When a vacuole informed me that he was the storage. So when you go inside a cell, remember what you see. There's over a trillion cells in both you and me. Just sing this song if you ever feel confusion. And remember, active transport is the opposite of diffusion.
All right. So that's just a fun way to start it. We're going to be talking about all of those components. Hopefully, you've learned about some of those before in other courses that you've taken, and we're just going to give you a little bit more detail. So to review, and it must have been hard to come up with some of those rhymes, eh? What can you rhyme with Golgi complex? Like you did a good job of that. We have plant cells that are rigid. They have big vacuoles in them, dense cell walls. We have animal cells, which are more circular, flexible, fluid. Um, they're what we have. So we're going to talk about our innards, our cells today. And then a little bit about bacteria. Most of the section on bacteria, the prokaryotes, is going to be your online study component for next week. Okay, so that's going to be what you need to do next week. So as we go through this, maybe on the back of your note paper or on another piece of paper, I want you to create a table. So I have kind of taken all of the components that we're going to go through and made a little table in the bottom corner. And I've just like helped you figure out, do bacteria, plants, and animals have this component to them? Because it's a really easy test question and it says, hmm, where am I not going to find a nucleus? Which type of cell? So then you could say, oh, well, of course, prokaryotes don't have nucleus, and you know easily just looking at this chart. Okay, so you'll see here in the bottom right corner, plant cell, animal cell, bacteria cell. No, it doesn't have it. Yes, it does. Yes, it does. Okay. So I want you to make that comparison table as we go through the lecture because I don't think that little table is in your handout slides. It's just something I've added to mine to kind of help organize kind of information for you. Okay. So you can kind of go through that as you listen to me yap on. It'll keep your brain busy that you have something to do and copy down. So we're going to start by looking at cell walls. Animal cells, you and me, do not have cell walls. We do have cell membranes, which we will talk about. They get their own little section in this lecture, but we don't have cell walls. Cell walls are really thick and they're made with cellulose or chitin, which aren't really as common in animal cells. Those are more structural components for those, like mushrooms and insects and plants and other things. They're really stiff coatings that are around the outside of the plant cell, or the fungus or bacteria, but mostly we could think of these in association with plant cells. And they give it a little bit more structure. I kind of visualize it like a wax coating on the cell. In my mind, that's how I see it because it helps hold its shape and keeps it in the right place, but it's also a little bit flexible. Not as much as a membrane is, but it helps kind of hold the water in. So when you think of leaves, some of the leaves have waxy coatings on plants, and that's to help control the water movement. And that's sort of where my vision of wax as a cell wall comes from. But it's made from cellulose or chitin, and plants can have multiple layers of these cell walls, but animals do not. Okay, so here's an example. Here's the cell in the middle. So inside the yellow circle here, the yellow circle is the plasma membrane. We have two different plant cell wall layers in this example. And then there's an actual connection between the two different cells. So these cells have been kind of cut in half, and we're looking at them from the top down. We have the light brown cell wall membrane, and then the dark brown cell wall membrane. The yellow is your plasma membrane, and then the components of the cell. Okay, so cell walls are mostly for bacteria and plants. It's an extra layer of structure. We're going to skip that one. Next, we're looking at the cytoskeleton. So here is kind of the something you've hopefully heard of before. Animal cells and plant cells do have cytoskeletons, but bacteria do not. Those prokaryotes don't need them. And think of the size difference between animals and bacteria cells, right? Animal cells are 10 times bigger. They're 10 microns or bigger, where bacteria cells are at most 1 micron. Okay, so it's a 10 times size difference. These different tubes and structures kind of work like a highway to move things around the cell. Okay, so there's the microfilaments, which are made of actin, and they help the cell move different organelles around, or the amoeba, when it puts out its pseudo arms to grab things, it uses that. The intermediate filaments are kind of smaller in between, and they help anchor things in place. Like the nucleus is anchored in place. You don't want it floating around and bouncing against other stuff, not if it's got your delicate DNA inside it. So you want certain things anchored, and they will anchor onto the microfilaments. The microtubules are made of tubulin, and these are straight hollow proteins that will kind of serve the function of both. So they will anchor organelles in place, but their most common use is when chromosomes divide during mitosis, which we will talk about kind of in the second half of the course. Okay, so here is a picture of a cell. This looks like it's a plant cell because it's kind of a rectangular shape. Okay, so we have the plasma membrane on the outside. Okay, these orange bands that are kind of running across straight up and down and back and forth, kind of lateral and horizontal, they are the microfilaments. And microfilaments are made of actin. Okay, so keep that. I always remember filaments has an A in it. A is the same letter that starts actin. That's how I remember actin makes microfilaments. Or microtubules, which are these big yellow lines here, much thicker, big yellow lines. Microtubules have tube right in the name, so they're made of tubulin. Okay. And then there's these intermediate filaments that are kind of going in diagonal directions across it. But if you notice the organelles here, so this looks like it's part of the endoplasmic reticulum, this pink thing with the black dots. Over here, this is the mitochondria, this orangish thing with the stroma inside it. They're attached to the microtubules because it kind of holds them in place. Notice how the microtubules are attached to those organelles. And the intermediate filaments here kind of go on the diagonal, and you've got ribosomes attached to them, and it can kind of help the ribosomes move around to wherever they need to be. because ribosomes kind of can float free in the cytoplasm or can move around. We also have a vesicle here that's moving around using this microtubule highway. So kind of think of it like these orange tubules that are running vertical and horizontal predominantly are like your 401 highways. And then you have your, city streets cutting it off on the diagonal. And then, your main regional roads as the microtubules. I'm just trying to give you another visual of how things can move around in the cell. Okay. So here's a real scanning electron micrograph of cells. And the microtubules are in red. So all of the glowing red you see are different microtubules spreading across the cell. And you can see they're kind of going in all sorts of different directions. They're going with the outreached tentacle areas here. They're going back and forth in the center area here. The blue is the microfilaments, so made of actin. And you can see them all over the cell as well. They don't really strictly follow one or two directions, but they do overlap quite a bit with the microtubules. And then the green dot in the center is your big nucleus. Okay, so they help hold things in place or shuffle things around like roads in the cell. There's different functions of the cytoskeleton. So if the cell needs to change shape, they are heavily involved in doing that. They help the cells move. So your cells aren't moving so much, but think of bacteria cells. Um, they don't have the strong cytoskeleton. They need another way to move. Uh, so sorry, bad example. Um, I was trying to link that to the cilia and the flagella that we talked about earlier. Um, those are structures that are going to like move along, say your trachea and your lungs. You're going to have little hairs and little fibers in your noses that are kind of beating in one direction. They're connected to the cytoskeleton in the cell, kind of to help things move. Um, they're very involved in the cytokinesis. So when two cells are dividing and you need to cut a new cell wall in between the two separate daughter cells now, um, the microtubules are very heavily involved in that and chromosome movement. So we'll see a picture. Um, where's my picture of chromosomes? Oh, the chromosome picture will come up later, where we can see the microtubules are involved in separating them out. Okay, so here's a practice question. If there was a drug that inhibited you from making microtubules, so your body cannot make these microtubules, what happens to the cell? What is going to happen? So here's where I want you to put in the chat what you think the answer would be. And this is a harder test question. That's why it's in the lecture for discussion and not just on the test. What do you think the answer would be? You don't have any microtubules. So think of what microtubules are involved in and then put in the chat what the cause of that would be. Not the cause, the consequence, that's the word. Okay, so we could have if there's no microtubules, the cell cannot move. There's no organelle movement within the cell. We cannot have the cells dividing. We cannot do that cytokinesis. Another option is that we prevent the formation of new cilia and flagella, so like little hairs that move. Um, Another option is that the cell would die if it doesn't have these microtubules in place, holding things where they're supposed to be or moving things where they need to be. The cell would ultimately die. Or we could say that all of that happens. So what do you think is the correct answer? Thank you, Jordan, for guessing. Okay, Michael's made a guess. Annette, good. I'd like everyone to make a guess. This is that 5-minute break in that Pomodoro method. It keeps you awake and doing a different task so that your brain can reset before we go into the next section. Okay. So you're all kind of correct. The correct answer is E. Everything does happen. So if we don't have our microtubules, we cannot divide. The cell can't divide. If the cell can't divide, it's going to actually die because it will eventually get so big, so big, so big. It'll duplicate its nucleus. It'll have double the set of chromosomes. And if it can't divide, it will get too big and then die off, that it can't diffuse materials and food from its outside area into where all of it needs to be. Okay, so that's definitely important. Organelles would stop moving, cell division would be inhibited, which would kill the cell, and you would prevent new cilia or flagella from attaching that have that waving motion. Okay. So the cilia and flagella here, um, we have those in animal cells, and bacteria have them as well, but the structure and the mechanism in the bacteria cell are a little bit different. They function a little differently. So they are given the same name, but they are operating in a different way. We're going to talk about the cilia and flagella here of animal cells. So eukaryotic cells, you and me. We're talking about your bodies here. So cilia are help to move the cells around or to help move things past the cells. So a lot of fluid environments in your body, you need to have that fluid moving past. So think of your digestive system. You eat something, you need to swallow it, move it into your stomach, move it through your intestinal system and out your body. So you need something in the body to help tell the things what directions to go, kind of like, your police officer saying that way, wrong way, a one-way street, go that way. So the cilia will help do that. Okay. Um, and we'll see pictures of kind of what that looks like. So the top here, this picture is the cilia inside your trachea, your lungs, your breathing tubes, and they kind of flap in the same direction. They kind of beat back and forth to help move things in the directions they're supposed to be inside your lungs. The flagellum that we'll see in eukaryotic cells is mostly associated with the human sperm cell. It's got that long tail that spins like a propeller that helps it move. The propeller motion that you'll see from a bacterial flagella is slightly different. So just note that they're same name, same shape, but different mechanism of movement. So this slide was just put here for a comparison. You don't need to study it because it's not in your handout slides. But just notice here that there's some major differences between the bacteria and the eukaryotes. The eukaryotic flagella are large and thick, and they contain microtubules. But the bacteria flagella are small and really, really thin, and they don't have those microtubules at all. So they have a different kind of motor that moves the motion of the flagella, helping it kind of move the cell. So next we have a nucleus. All cells have genetic information, but only the plants and animal cells have it contained within the nucleus. The bacteria just have it kind of free floating in a quarantined area of the cytoplasm. So the nucleus is that large circular thing we've seen at the center in our pictures. There's a nuclear envelope that kind of like a balloon around the outside of it. It's got little holes in it so things can come in and out when we're making new proteins. It's got chromatin versus chromosomes. We'll talk about that. And then it's got a nucleolus in the center. So let's look at kind of the pictures to see the distinction there. So here's the picture. We have our nucleolus in the center, which has all of the dense chromatin in it. Okay. We have our nuclear envelope around the outside with these holes in it, these nuclear pores. Okay. These pores are designed so that it allows the RNA, which is used to make proteins, to get in and out of the nucleus because it has to go out into the endoplasmic reticulum, which you can see in the picture here is attached to the nucleus. The endoplasmic reticulum is involved in making those proteins, but it needs to get the instructions on how to make the proteins from the DNA. So there has to be a connection, and that's what these nuclear pores are for. Okay, so the chromatin that is out here in the nuclear space is usually chromatin that is being copied in some form or another. So the RNA or the DNA polymerases will be in there single-stranded, making it single-stranded so that you can copy the nucleotide sequence. Okay, so let's go back here because I skipped this one. So the nuclear envelope helps separate the chromosomes, the chromatin, from the cytoplasm. We don't want it free floating everywhere because the genetic information to build a person is significantly more complex than it is to build a bacteria. We need to have a strong organization system, and that's where the nuclear envelope comes in. It helps keep the DNA contained and organized in a way that we can quickly access the specific gene that we need and not have to go through everything else. So we talked about why there are perforated holes so that we can allow different small molecules to pass through when they're replicating. Um, the DNA or when they are making a protein and reading off of it. Okay. So the DNA, which is our genetic information, it's configured in two different forms. We can have a compacted chromosome, which is the thing you see on TV a lot that they show with like the X shape kind of thing. But we only see those during cell division, that they're that structured and that formatted, because we know we have a copy of every single gene when it's all tied up tight into that chromosome structure. If it's just the regular cell growth phase, we don't need to have it so tightly confined because we're going to be copying and making proteins from different parts of that same piece of chromosome. So we're going to need to have access to lots of pieces out at the same time so it doesn't form up into that structure that we see. So that we would see more of a chromatin, like single threads versus a spool of thread. So we have the nucleolus that we can see where the ribosome synthesis happens. So we have to make the ribosomes, transport them out of the nucleolus to the endoplasmic reticulum so that they can actually be used there to then synthesize proteins. Okay, so here's the picture where we have this pink here in this one cell is the chromatin. Okay, so you can see the nucleus here. This is the nucleolus in the center. And it's very spread out and equally kind of pink, right? This next cell beside it that's dividing, you can see it's very clear fingers. This is the chromosome because we need to make sure we have the equal copies of this chromosome on this side going to one cell and the same chromosome on the other side going to the other cell so that they show up differently. So chromatin is just single, loosely floating around in the nuclear envelope, where chromatin is formed into those chromosomes are tightly wound structures that we would recognize. Okay. Now it's time to keep you awake and play a game. So this is the first time I've used this platform to do sticky notes. So we're going to go over to, hold on, I'm going to give you the link in the chat. We're going to go over to a Canva page. So you can scan the QR code there, or you can click on the link in the chat. And we're going to go over to another page and start putting the information in that we know. So I'm just going to switch over again to share the right thing. Okay. So good. I can see some of you are coming in here. So you should be able to click on the sticky note option at the side, choose a color of sticky note that you want. It doesn't matter what color it is, just have fun with it. How do I move it? There we go. And in your sticky note, someone's typing in my sticky note, and that's okay. In your sticky note, you're going to tell me one organelle that we learned about, what its function is, and the types of cells that have that organelle. It's okay. You don't have to erase it. Yeah, so just move your sticky note somewhere else, choose a different color if you want. Yeah, so tell me what your organelle is that you're going to talk about, what its function is, and what types of cells, animals, plants, or bacteria, have that organelle in it. So you can all pick, yep, pick a sticky note, change any color you want. It's anonymous, so you can kind of change it up if you want. And you're allowed to use the information from your notes. So you can write about an organelle that we've covered this morning or one that's coming up later in the lecture. Just want to use this to make sure everybody's still with me and awake and engaging. So we've got the nucleus, I see, is being done. So tell me which organelle you're going to type in, what its function is, and the type of cells that have it. So the link is in the chat for you to get to this whiteboard that we are playing on. Okay. Good. So I only see two of you typing. Where are the rest of you? Okay, three of you. I have three. I have mitochondria, ribosomes, and nucleus. I know we've talked about other organelles, or there's more organelles in the notes you can look ahead on. Let's see. I'd like to see everybody making one post-it in this whiteboard. That's my goal. Lots of different cell organelles we can talk about. So I have mitochondria, ribosomes, and the nucleus. Is anybody else going to add in here? Pick an organelle. Could have been one from the song that we started out with. Went into a cell to get out of the rain. I listened to it a little too much this week. Okay. Whoever is the tree frog, just pick one organelle, and you're going to tell me what the function of that organelle is, and you're going to tell me what types of cells have it, plants, animals, or bacteria. Okay, good. So we have Golgi apparatus, ribosomes, mitochondria, and nucleus. And where's everybody else? I'm not here. Okay. Good. Looks like we have cytoplasm coming up. Awesome. So pick an organelle. So the Golgi, whoever did the Golgi, my tree frog person, what is the function? You've got it. What organisms have a Golgi? Plants, animals, or bacteria? Okay, chloroplasts, excellent. What's the function of a chloroplast and what types of cells have it? Good. Just going to move this mitochondria down a little. Okay. Good. So this is kind of testing your brain. What's the function of the chloroplast? We haven't covered that yet, so you're working ahead. Or you're going off of former knowledge, which is awesome. Good. Good. Two, three. So that's 6 of my 9, 10 students. Okay. Well done, those of you who participated and engaged. I appreciate that. It'll benefit your learning, I promise. When we get to the test, you'll be like, oh yeah, I put that on the whiteboard. I remember now. Awesome. So chloroplasts are responsible for photosynthesis, and they are only in plant cells. There are some algae that do the photosynthesis as well. So the prokaryotes kind of, yeah, have the chloroplasts. A little bit. But good. Well done, everyone. Thank you for participating. So let's go back to our lecture. Oh, we got cytoplasm, gel-like substance within the cell. So it sort of maintains the fluid shape and structure of the cell too. Yep, plants, animals. Do bacteria have cytoplasm? So that's a good question. Actually, I think I might even use that as a practice question. What organelles do all prokaryotes and eukaryotes have? Because it's not as big a list as you think. It's actually a small list of what everybody has. So great. Okay, let's go back to the lecture then. Everybody is still with me, is sort of the message I'm taking from that. Um, okay. So next, we're going to talk about systems of membranes. So here's where we get into the plasma membrane. So plasma membrane is something that every type of cell has. Bacteria, plants, animals, it doesn't matter. They all have a plasma membrane. It's one of the few that is consistent within everybody. Okay. So the plasma membrane is made with those lipids. So if you learned, um, you did the online work for week 2, talking about lipids, it should have mentioned a phospholipid. Those are what the plasma membrane is made out of. Okay, so there's one side of it that water is water loving and one side of it that is water fearing. Okay, so they are used to isolate the individual cells and help control what gets in and out of the cell. So they're kind of like the door where you decide what can and can't come through the door to your house is what the plasma membrane will partly do. It would be with inside the cell wall for those bacteria and plants that have cell walls. The cell wall would be on the outside and the plasma membrane would be on the inside. So this is a very typical picture of it's called a fluid mosaic model of the plasma membrane. So here we have our little phospholipids, this little circle with 2 legs. That's a singular phospholipid. And they line up with their legs together. They're like touching feet because their feet are hydrophobic. They're afraid of water. They don't like the water. So the cytoplasm inside the cell is something that's uncomfortable for these little tails of the phospholipids. So they line up in a way where the heads here block off all of the access to the liquid. So the cytoplasm liquid inside the cell, the watery environment outside the cell isn't going to contact the center because these heads, called hydrophilic, meaning water loving, heads will block the water from getting through. So notice how they're very tightly packed together here. Things can't just wiggle their way through the phospholipid membrane. It's very tightly controlled. You need to be able to get through in a certain way, and that's what some of these blue things are in the picture here. These are protein channels. So they're very specific to certain things. They're like, I will let you go through, but I won't let you go through. So they kind of control who can come through here. And that's important because it helps control um, like the trigger pressure inside the cell, the ion balance inside the cell, and a lot of the other chemical reactions going on inside the cell are controlled by what's in and out of the cell. Over here, sticking out of the cell, we have something called a glycoprotein. So it is a protein, but on the end of the protein, these little circles here, this chain of circles is made of carbohydrates. So that's what the glyco stands for. Glyco means sugar. So protein with a sugar attached is a glycoprotein here. And these are things that could be used by like your immune system. So your body would go around and recognize this pattern of sugars, the shape of the sugars on the outside of this cell, and it'll be a message to the immune system saying, nope, this is one of our cells. We're not attacking it. But if it sees a sugar pattern that is different, it says, oh, wait, that's not me. That might be a bacterial cell. I'm going to attack it. So they're kind of messages for the immune system, and you can see different glycoproteins here. We can also have glycolipids, so carbohydrates attached to the phospholipids head, so that we can kind of give signals to the other cells in our body, in our system, that kind of say, this is what's going on, okay? So we can see underneath the cell, the bottom of the cell here is your cytoskeleton. So these would be your microfilaments and microtubules. Your phospholipid bilayer, because there's 2 layers pointing in towards each other so that the tails of the lipids are together and the heads are outside, blocking off the aqueous environment. So the membrane is important because it helps control what can go in and out of the cell, but it's also used to move things around within the cell. So when we have something, say we make a protein in the we start making a protein in the nucleus, but that protein needs to be folded into alpha helixes, and it needs to be added to other protein subunits to make its quaternary protein shape. We talked about that the other day. We would move it around inside these things called vesicles. So this is just like your phospholipid bilayer on the outside of your cell, just it's a really small version of it. It's like a little bubble inside the cell that can either join to the outer membrane and release things, or it can just move to another part of the cell, join to that, and then share what's inside it here. So this can be carrying good things, it could be carrying waste. It could be carrying different chemicals around that need to move throughout the cell. So part of this membrane system is your endoplasmic reticulum. So these are what's attached to the nucleus, and they're all membranes. They're all these phospholipid bilayer membranes, and they're used to help give a lot of surface area for the ribosomes to sit on and those proteins to be made, because without proteins, you'd be kind of dead. They're really important to a lot of functions in our body to keep us alive. So we have to have a lot of surface area, a lot of space to make these proteins. And some proteins can be really, really complex, that you can't just put a bunch of amino acids in a line and say, go do your job. After you've put the amino acids in a line, they need to be folded, then they need to be edited, then they need to have something cut off of it, and then something added to it. So there's a lot of different steps that are going to happen to this protein as it moves through these two endoplasmic reticulums. So we have the smooth endoplasmic reticulum, and it's just called smooth because there is no ribosomes attached to it. It's just the membrane. The rough endoplasmic reticulum is the exact same as the smooth endoplasmic reticulum, but it has those little ribosome dots attached to it, which is the site of protein synthesis. So the ribosome is where the different amino acids would be added to the chain of amino acids to make your protein. Okay. So if we go back and look at this picture again here, we can see attached to the nucleus, we have this bilayer of membranes, and all of these black dots, these black circles, are your ribosomes, where the amino acids are being added to the chain of amino acids to make the protein. And you can see them all lined up along there. And then, so we start with the rough endoplasmic reticulum with all these ribosomes. And then we come out here to the smooth endoplasmic reticulum at the end where it doesn't have any of those black dots. And you can see these little bubbles, these little, looks like bubble gum, doesn't it? These little vesicles popping off of the end of the smooth endoplasmic reticulum. And now this protein has all of the amino acids that it's coded for, but it still needs to have something else happen to it. So it's going to be moved to a different location in the cell. And then we have these scanning electron micrographs here where you can see here, the yellow line here is the lipid membrane, and all those red dots are the ribosomes that are making proteins. Okay. The smooth endoplasmic reticulum doesn't have the color, but it's got these little circles, these little bubbles, these holes. Those are all of your vesicles that are moving to different places in the cell. So later in the course, we're going to have a much more in-depth look at the process of making proteins, and we'll get a little bit more detail. But generally, there's kind of two paths that the protein can go on. So first, you get RNA starting in the nucleus. That RNA comes out and it goes to the rough endoplasmic reticulum, where the ribosomes will add the amino acids to make the protein. It can go into a vesicle from the rough endoplasmic reticulum to the smooth endoplasmic reticulum, or it can go directly from the rough endoplasmic reticulum to the smooth endoplasmic reticulum. That's what my arrowing on the bottom here means. So this corner here can pop off its own little vesicle, and then you might have a little vesicle floating up to the smooth endoplasmic reticulum here. It doesn't need to go through all of these little hallways here in the rough endoplasmic reticulum to get to the smooth one. It can bypass it. So it can maybe, this ribosome here is finished its protein. It's probably going to make a vesicle that pops off of the rough endoplasmic reticulum and moves to the smooth endoplasmic reticulum. So it will fuse, modify the protein, and then it'll let it go again. Okay. After it leaves the endoplasmic reticulums, it's going to go to the Golgi body, which will do more manipulation of the proteins, and then eventually to the plasma membrane to be released from the cell. Okay, so the smooth endoplasmic reticulum has no ribosomes. We know that. The rough endoplasmic reticulum does have the ribosomes, and they have slightly different kind of areas of specialties. So the smooth endoplasmic reticulum has lots of enzymes. So here is where, like in the liver, you would detoxify your drugs. So if you take aspirin or take too much Advil and it's damages your liver, it's because you're doing too much detoxifying with your smooth endoplasmic reticulum. The rough endoplasmic reticulum produces a lot of those proteins because that's what the ribosomes function as, and it makes the phospholipids that are going to the outer phospholipid membrane of the cell. It would kind of replace the old ones that may be dying or just getting into trouble. Okay. So next we have in our system of membranes, we go to the Golgi apparatus. So again, animals and plant cells have these functions. Bacteria do not. Bacteria are so much smaller, they can just have the ribosomes floating freely in the cytoplasm and still, bounce with everything and encounter the ingredients that are needed to make its own proteins. So the Golgi is a set of stacked, flattened sacs, is how it's described. So I kind of think of like smushed pancakes. When we see the picture, that it might make sense. And it will get these little vesicles coming from the endoplasmic reticulum that will go into the Golgi and it'll bond and sort of mold with the membranes of the Golgi. And then it will modify the proteins again some more. So here is where the sugars, the glycoproteins, are created. They're adding the sugars to the amino acid chains, and then it'll have a different function after it's folded and ready to go. Okay, and here's where things get finally packaged into the final vesicle that will take them out of the cell. Okay, so we can see the Golgi here. Excuse me. It's not attached to the reticulums, but it's close to the endoplasmic reticulums. And things flow in a singular direction through them, just like the endoplasmic reticulum. So if this vesicle here with the blue circles is proteins coming in to be modified, it will fuse with the Golgi apparatus. And then different things will be added to it. Different sugars might be added to it. Different things will be cut off of it. And as it moves through this membrane, like through these sacs of membranes, it will change shape, function, activity, and that's why it's depicted by different colors. So when we're finally finished here, we get these orange proteins now that are packaged in this little vesicle, ready to go and do what their job is to do. So they might bond with the plasma membrane and release it into the external environment of the cell. They might go and somewhere else in the cell where they're needed and do whatever their job is. Okay. So the different proteins or substances that are made in this membrane system that we just talked about. So endoplasmic reticulum, Golgi apparatus, and plasma membrane. Those three are all kind of all membranes. They will do three different things. The first one is they'll make proteins that are secreted outside of the cell. So that could be something, I'm just trying to think what kind of proteins we secrete. Ooh, biology. I'm thinking like nursing lesson for me. I can't name a protein that was cells secrete. Can anybody name a protein our cells secrete? Off the top of my head, it's not a normal example isn't coming to my mind. So let's look at the figure here. Oh, amino acids aren't the same as the proteins. They are the start of a protein, but not a final protein. So you've got the right idea, Michael. Antibodies for proteins produced by the white blood cells. Here's where this example comes back. So let's look at the picture here. We have our nucleus at the bottom left. We are going to take RNA from the nucleus. We're going to give it to the ribosomes here on the rough endoplasmic reticulum. It's going to build the amino acids by putting one after the other into the long chain of amino acids. That's what these red Ys are. They are going to be packaged in a vesicle here off of the smooth endoplasmic reticulum. It's going to travel up to the Golgi apparatus and bond there. The membranes are going to fuse together. And then it's going to be manipulated and altered within the Golgi apparatus, and that's why the color is changing here. So here we have a finalized antigen. So this would be something that is your body has recognized there's a foreign thing, you've got a cold in your body, and it's going to make a protein that's got the shape of the virus so that your body knows to attack that thing. So it's making a protein here and it's putting the shape of the virus on the outside of the protein, and it's going to take it to the plasma membrane and just release it into the body. So now your body knows that if I see lots of these things attached to a cell, they're really bad. I'm going to go and get rid of them and kick them out. I'm going to use exocytosis, which is part of the online learning to learn that word. And I'm going to spit them out of the cell. So that's one function of these membrane systems is to produce secreted proteins. So an example there was the immune system. The two other functions, one is digestive proteins are made in the rough endoplasmic reticulum. They travel to the Golgi. They're packaged there as lysozymes. So that's a word where there's lysozymes will break down different things, kind of like stomach acid. It'll break down things into smaller parts that your body can use. So the lysozyme is like the stomach acid that will fuse with the food vacuole. So wherever the food is in the cell, the lysozymes will fuse with it, break that large chunk of food down into the individual chemical components of it that your body can reuse for other actions. Okay, so that's a second function. So let's look at that picture before I talk about the third. Same process. We have our nucleus giving the instructions on how to make a digestive enzyme. The RNA comes out here, the ribosomes pick it up, they make the amino acid chain that relates to the enzyme. It gets moved through these membrane systems and it leaves the smooth endoplasmic reticulum as a vesicle. This vesicle moves up and it bonds with the Golgi apparatus, which again modifies the enzyme, modifies that protein chain or structure, adds sugars to it, whatever is needed. And then these vesicles stay inside the cell, but they will bond with food vacuoles. So out here, this is a little pocket that the cell is grabbing some food from the external environment and bringing it in. It's like eating, right? It's eating some food. And now the lysozyme proteins, enzymes, are going to bind with the vesicle that has the food in it and break that down to its individual chemical components that can be released into the cell for use. Okay, so we have two vesicles, one coming from the external environment and one coming from the Golgi, and they're going to bond together and get rid of our food or use the food, break it down. The third function of these systems of membranes is that they are going to make the membranes that are going to replace what's old and damaged. Okay, so the endoplasmic reticulum needs to be replaced. The plasma membrane may get damaged and needs to be replaced. So freezing something and thawing it out will poke a lot of holes. The ice crystals will poke a lot of holes into the plasma membrane, so that needs to be repaired. And the third function of these membranes is they will make these phospholipids or other components that will replace them. Okay. So let's do some practice questions here. An organelle that is associated with carbohydrate synthesis in eukaryotic cells is what? Carbohydrate synthesis in eukaryotic cells. What part of the system of membranes that I've been talking about relates to carbohydrates? So we have the Golgi complex, the ribosomes, the centriole, the nucleus or nucleolus. So take a guess and put it in the chat, please. Okay, we have two guesses. Anybody else want to guess before I tell you the answer? Good. Everyone's in agreement so far. Carbohydrates are added or adjusted in the Golgi complex where they are put on the final protein. So you're all correct. That's right. Okay, so here's another practice question. Why are cells so small? They like being invisible to the naked eye. They need to be able to move fast when a predator comes. They need to be able to quickly transport molecules to the center of the cell. They can be bigger if they had more cytoskeletal features, which is correct. So A, B, C, D. So everybody take your guess in the chat, please. I have three guesses. Good. Four guesses. Good. And everyone's in agreement, and you're all correct. Cells need to be small because they have to move things in and out of the cell to the center of the cell, right? To get things to the nucleus and all the supplies that are needed for the cell to function. So if it's too far of a distance, your little vesicle is going to run out of gas and not get there if it's too big. Good. All right, so let's look at some more organelles and features of cells. Vacuoles, which is oh, it's another one that is in all three cell components. Who remembers what else there was in all three cells, in all three cell types, plants, animals, bacteria? What was the other thing that was in all three cell types? Who remembers? Cilia and flagella. Nope, plant cells don't have cilia and flagella. Good guess, though. Animals and bacteria do. Starts with a C. Cytoplasm. Every cell needs to have cytoplasm. Did we cover that yet? I think we did. Okay, so we're going to talk about vacuoles. So we've learned a little bit about those vesicles that move through the membrane system of making proteins. Here we're going to talk about vacuoles, which are similar. It's a membrane that is around a certain um, function. So it's going to be a circular bubble that has a certain function. So there's different kinds of vacuoles. We can have contractile vacuoles, which means they squish, they squeeze, they contract themselves. So a lot of little algae will use these. They'll fill them up with water by osmosis, and then they'll contract. And as they squish the water out, it'll propel them forward in kind of the water. So one type of vacuole you can have is a contractile vacuole. So here is a contractile vacuole in a euglena, and it's full of water here, and the bacteria or the euglena will empty it by contracting the vacuole, and it's empty here. There's nothing in it, but now the euglena has moved. It's propelled itself through the water with that motion. Okay. We can also have plants have central vacuoles. So these are used to kind of keep the shape of the cell to help keep it square and rectangular, to store hazardous wastes, and to also, be extra fluid for the cytoplasm to keep its trigger pressure high and in the right balance. Okay. So it's used to just store water that the plant could need. Because if a plant doesn't have water stored in its individual cells, it's not going to photosynthesize very well, right? So we have contractile vacuoles. Plants have central vacuoles. I feel like we skipped a slide here. Nope. Okay. Just two kinds of vacuoles. So next is mitochondria. Okay, so mitochondria, I'm sure you've learned about in, your previous studies. They're called the powerhouse of the cell, the energy center of the cell. And we're going to go into a lot more detail later in the course. But this is where the cell generates its energy. This is where it makes ATP. It takes that glucose molecule and it's going to break it down into the individual carbon pieces and take the energy out of all of those bonds. Okay. So it has different structures to it. So here we'll look at this. We have an inner matrix area where we're going to pump this full of hydrogen ions so that it's so full it's exploding, and it's going to force them out with a lot of force. Like, think if you blow up a balloon and you hold it there and you pinch it, when you let go of the balloon, the air forces itself out and it just moves around the room, right? We're going to fill up the center of the mitochondria here with hydrogen ions, and then they're going to be forced to go back through this membrane to get out because there's so many of them. So this inner membrane here, these folds are called the cristae, and we need a lot of surface area because that's where the hydrogen pump is going to capture the energy that makes the ATP. So we're going to push the hydrogen in, it's going to force itself out back into this intermediate area here, intermembrane compartment between the two. Okay, so it's nice and smooth on the outside. It's like a little oval shape, but the inside, these cristae are folded and loopy, so we have lots of surface area to work our protein pumps that are going to make our ATP. Ooh, we have a YouTube video. Let's see if we can watch this one. Oh, I'm going to get that video. Just give me a sec. This is cool to watch videos, right? All right. Can't open it. What's wrong? Oh, okay. Sorry, no videos. I'll have to check on that link for you guys. Okay. So let's go back to our presentation. There are videos in the online component. I can link some videos up there for you. Okay. So here's a scanning electron micrograph of the mitochondria. And it's not exactly the way the picture draws it, but you can see it's smooth outer edges. And then all of these little lines inside here are those folds in the cristae, where we're creating surface area so that we can pump hydrogen in and out of those matrix area in the center here. Okay. So we know that the mitochondria is where we get the majority of food from. Um, but it's a really cool theory that mitochondria themselves used to be free living organisms, that they didn't really um, need to be inside another cell. They lived outside of the other cell. And then the other cell said, well, if you make so much energy, it's advantageous that we live together. I'll give you a safe spot within my cell to live if you keep making all this extra energy that you don't need and share it with me. So it's an endosymbiotic hypothesis states that the mitochondria used to be their very own living organism because they make all their own energy. They have their own DNA. They have separate DNA from what's in the nucleus, and they can extract um, energy either aerobically or anaerobically. It doesn't matter if there's oxygen present or no oxygen present, they can still kind of work. So that was a really cool like thing about the mitochondria. So a lot of times um, we can track people through their mitochondria. So which parent did you get your mitochondria from? Do you know? If you've ever done those like 23andMe or those DNA, what was the other one? Can't remember the name of the other one. Yeah, and that's right. You get it from your mom. Your mom gives you your mitochondria. You can't get any mitochondria DNA from your dad. It doesn't happen. So one of the neat things that they can do is they can trace your genetic, your chromosomal DNA and see where your family line is from. And that's generally a mix of both parents, right? Your chromosomal DNA is a mix of both parents, and you can trace it back to see where it comes from. But they can also strictly trace the female line. You can trace all the females and not worry about your dads at all and just see where the mitochondrial DNA can come from. So this is an interesting thing when you have people being like surrogates or like cloning. So when you have things being cloned, you're going to take an egg from a person or an animal. You're going to take the nucleus out of that egg. So you're going to remove all the nucleus DNA, all the chromosomal DNA, but you're not going to take the mitochondria out. So when you add DNA from your cloned species, so I want to clone sheep X, I'm going to take the nucleus out of the egg from sheep X, or in the case of Dolly the sheep, the first cloned animal, they used an udder cell. So they took like a skin cell, they took a nucleus out of the skin cell, they put it into the egg of another cell, but that egg has mitochondrial DNA from the original donor of the egg, not the same as the chromosomal DNA that was taken from the skin cell. So you can grow these, it'll still work. You'll still get a live animal, obviously. That's why how we got Dolly the sheep. But Dolly the sheep was interesting because she had two different sets of chromosomes, and she wasn't like a 100% copy of her parent, where she came from, because the mitochondrial DNA was different, right? And that might have an impact on why she didn't live as long as her mother, right? Not her mother, I shouldn't say that. The sheep that she was cloned from, Dolly didn't live as long as the original sheep where they took the skin cells from, because her mitochondrial DNA came from another sheep. And I don't know how old that sheep was, but it's just neat to know that, right? So when you're cloning something, you got to think about all the DNA and where it's coming from. Mitochondria have their own DNA. So if you're going to use an egg or any cell that you're going to transfer DNA into, you're also going to transfer that. Why they don't transfer the mitochondria at the same time, I'm not too sure. Maybe they're just too small that they can't. Um, take them out without damaging them properly. I don't know. I don't know that yet. Science will figure it out, I'm sure. But it was cool. All right, so let's keep moving on because this is on the test next week, so I got to cover it today. So, chloroplasts, which somebody put in our whiteboard on a post-it, was awesome. Chloroplasts are the specialized organelles in mostly plant cells, but some algae, prokaryotic algae, have these as well. And they're responsible for photosynthesis. So they kind of look like the mitochondria, where they have a smooth outer surface and then a lot of surface area inside of it. And those are the grana, which are made up of thylakoids. So let's look at the picture to see how that works. So you have a smooth outer surface for the chloroplasts. And then you have all of these like stacks of pancakes, these little discs inside here, which become, as a term, they're called the grana. And each individual little pancake or disc is called a thylakoid. And in here, this is the location of photosynthesis. So this is where the sun will take, or the plant will take the sun and the carbon dioxide and the water and turn it into something like starch or glucose, and make their own energy stores from that. Okay, so we're not going to explore this reaction in as much detail later in the course, but we will look at aerobic respiration that happens in the mitochondria. Okay, so chloroplasts will take the carbon dioxide, water, light, turn it into glucose, and release the oxygen. So basic photosynthesis. This is the predominant location of it. And a lot of the chloroplasts have different color pigments. So predominantly we see green, right? Did you know you can drink chloroplast? Like there are companies that sell it. I remember I must have been like 18 or something, and my one of my aunts had started a one of those multi-level marketing things, I'm sure, but it was selling like natural health food stuff. And one of the things she sold us was chlorophyll. She's like, drink it. It's so good for you. Just like squirt it in water, like a little flavoring in a glass of water and drink chlorophyll. And it tasted like mint. It really did. And she's like, it's so good for you. Just drink it. Just drink it. And I used to drink chlorophyll, just straight up chlorophyll. It was weird, but, and then the bottle emptied and I never bought more. But yeah, you can drink chlorophyll. Never thought of that before, but you can. Sorry, I'm just trying to, I gotta move my, whoa. Okay. So next is what about leaves in the fall? We see different colors, right? So the green leaves are the ones that we see the majority of the year round because they have the highest amount of chlorophyll. And chlorophyll is the chemical that helps in the photosynthesis, right? But under that green, there are also carotenoids and other different types of chlorophyll that give you your yellow and red. So in the fall in Canada, the first thing you see is the green color will disappear from the leaves because the green chlorophyll, the plants are slowing down for their winter hibernation, right? Well, they don't hibernate, but their winter slow time. So all the chlorophyll is draining from the leaves, and once the green chlorophyll is drained, you get to see the yellow chlorophyll. And then the leaf will die because it's not photosynthesizing as much anymore because the plant is storing all of that energy. So that's kind of neat to see the different leaf colors. And this is just showing you the chemical structure and how it differentiates of those chemicals that will do the photosynthesis for you. So you have a chlorophyll here. It's a big ring structure with magnesium at the center. Your carotenoids, which are linear structures in the orange and the reds. And then the anthocyanins are your red colors here in your double ring structure. So just if you like chemistry of the environment, there you go. All right. So we have one last thing. I hope this is the last thing. Plastids. Plastids are only in plants. They're not in animals, and they're used for storage. So they can surround, sorry, the starch that is made from photosynthesis will be stored in these plastids for when the plant needs energy later. So say maybe it's nighttime and the sun is set, photosynthesis isn't happening as rapidly. You're going to need your energy from another source, so it would take it from these starch reserves that are held in these plastids. Okay. And you can also store different pigment molecules for various fruits, so get them to different colors as they're ripening up. So the chloroplasts and the plastids are usually free floating in the cytoplasm, but the plastids are just big globs here. And the only thing inside it is starch molecules. And remember, starch is just a long chain of individual glucose molecules. So there's lots of little things going on there. Okay, so let's do another practice question. Which of the following is or are evidence that mitochondria and chloroplasts were once free living organisms? Ooh, chloroplasts too. Hmm. They produce ATP, they contain their own DNA, or both capture energy from the sun and make ATP. So what do we think? A, B, or C? Or a combination of A, B's, and C's? We have a couple guesses here. Let's get a few more guesses before I tell you the answer. Okay, we've got B's and D's. Let's get some more. We have 3 B's and 1 D. So let's get another guess and see if we can get a little more info. Okay, another D. The correct answer is D. They do produce ATP. That's a correct statement. We know that mitochondria have their own DNA. So B is a correct statement, but so is A. Chloroplasts produce, oh, they don't produce ATP. They produce, oh, brain, think now. They do have to produce ATP a little bit. So they do produce, chloroplasts do produce a little bit of ATP in the production of the glucose. So D is the correct answer. A and B are correct. C is incorrect, which I think everybody caught that, because they both don't capture energy from the sunlight. The mitochondria doesn't get its energy from the sunlight. It has to get it from another source. Okay, so here's another practice question. If a plant mutates that has no chloroplasts at all, what would be the outcome? It would not be a green plant if it doesn't have chloroplasts. It would still be able to convert carbohydrates to energy. If it didn't have any chloroplasts, would it do that? If it does not have chloroplasts, would it still be able to capture energy from the sun? And would it synthesize new chloroplasts? Or more than one of the above is correct. So let's see, what do we think here? Just take a guess. Doesn't matter if you're right or wrong. So what's the function of the chloroplasts? That's where we hold the photosynthesis mostly happens, right? Okay, Dabby made a guess. Good, thank you. Michael guessed. Good. Anyone else? Okay. A lot of you are saying A. All right. Okay. The correct answer is E. There's more than one statement that is correct. A is one of the correct answers. They would not be green. That's true. But B is also correct. They would still be able to convert carbohydrates to energy. They would still do the photosynthesis, just not as efficiently. So they wouldn't completely die. Um, but they would still be able to convert carbohydrates to energy. So see the wording? They're not converting sunlight into energy, but that leftover starch that the cell has, it can still use that remaining starch if it didn't have a chloroplast, but it wouldn't live for very long. So A and B are correct. So that makes E the overall correct answer because both A and B are correct. Okay, let's practice another one. All cells possess all of the following components, except which do cells, so I'm talking plant, animal, and bacteria. What do they all have? Or which one doesn't belong? Which one is not in all three? Maybe I should word it that way. Okay. Couple guesses. Good. Thank you. So we're saying D, that that plasma membrane is not in a plant cell, an animal cell, and a bacteria cell. D is not the correct answer. D is not the correct answer. Think of the prokaryotes. Those are your bacteria. What does their name mean? Yeah, there you go, Christian, you got it. Okay, so all three cells have cytoplasm. All three cells have genetic material. All three types of cells do not have a nuclear membrane. Bacteria cells do not have nuclear membranes. They have genetic material, just it's not combined in a membrane. It's just free floating in the cell, okay? Plasma cell membrane, yes, they all have that, and they all need ribosomes. So nuclear membrane was the outlier because prokaryote bacteria cells do not have them. All right. So here, that table that you were supposed to make throughout this class, here's where I want you to use that table. If you made it, it'll be much easier to answer this question. Name one organelle that is only found in plant cells and nowhere else. There's two correct answers here. So tell me, what is the organelle and what is its function? Just choose one of the two correct answers. So you should have your table that says it is not part of a plant cell. Oh, plant cells do have cell walls, Annette. Yep. So check your table. So chloroplast is one. Actually, you know what? Maybe it is the only one. No, I'll accept a second answer. Let's see what you guys can come up with. What is an organelle that only plant cells have? No one else is sure? Yep. Good, Michael. Chloroplast is definitely one of them. So they are the site of photosynthesis. The second one I would accept is a central vacuole. Okay, there is also other types of vacuoles, but they're not central vacuoles. Those are specific to plant cells. So that would be the second one I take. Okay, same question, but with bacteria. What is unique to bacteria cells that other types of cells don't have? This might be a bit challenging, so let's see what you come up with. Take some guesses. What's different or unique in bacterial cells that's not in the others? It might not really show up in your table, though. Anybody want to guess? We haven't really taught you too much about bacteria yet. That's coming next semester. I'm looking forward to teaching that class. All right. So the answers, nucleolus, no. Bacteria cells don't have a nucleus. So the nucleolus, which is in the center of the nucleus, wouldn't be part of it. I have flagella. Their flagella function differently than the flagella of animal cells. So it's the same name, but a different function. So it's a tricky one. I understand that. And then I also said plasmids. So plasmids are small external pieces of DNA that are like circles, like a donut. Think of like a little donut as a plasmid. That they're free floating in the environment, or the bacteria can trade their DNA that way. Humans don't trade DNA the same way bacteria do. Oh, you were thinking of plasmids. Okay, good thought, Michael, then. All right, so let's try animal cells. What do animal cells have that the other two do not have? Oh, now I have to think. Actually, let's see. What do you come up with for this one? What does your table tell you? in your table where you said yes, yes, no, whatever that I was going through there. What do animals have that plant cells and bacteria don't have? Any guesses? I actually have to go and check my table because I'm second guessing myself now. The answer I have, what I was thinking of was cytoplasm or cytoskeleton. But here it says some plants have cytoskeletons, but it's not a predominant feature in all plant cells. So when we look at the plant cells here, we don't see the cytoskeleton, but we don't really see it in this animal cell very well either, in this graphic drawing. So I was thinking of cytoskeleton. That was the answer I was considering, but it might not be the best one. Okay. So you have hopefully made this table for yourself. For learning, I'm just going to make this big so we can see it for a minute. One of your online learning modules that you should be doing before the test is about prokaryotes. So I was going to suggest you make a comparison table like this to study from. So the definition of a eukaryote versus a prokaryote. What is cell size? How does it differ? What is the nucleus? Is it present or absent? If you can answer some of these things, do they both have ribosomes? What organelle does one have that the other doesn't? Kind of make yourself a comparison table as a way to study that. Let's go look at that module for online work then. So I need to share something different, that's all. All right, so this is your course. If you go into the lecture material here, the first thing is your online work for weeks 1 to 5. So cell transport would relate to the lecture I finished today. Okay, so that would be your online learning less like topic from cell transport. And then prokaryotes would be next week's online learning before the test. Okay, so biological molecules, you're going to learn about lipids. Cell transport here, this relates to one of the labs you're doing. I'm not sure if you've got into your uh, permeability lab yet, but this will be where all of the answers come from, this online module, okay? So I put a little review in here about the types of protein, like just of what we've learned about online, and then some practice questions for you, and then your independent study learning module starts here. Okay, so you're going to learn about the crossing cell membranes, how things move across cell membranes, the different types of ways to move things across the cell membrane. And I'm pretty sure you should have seen some of this before in your other education. So it shouldn't be new topics. It's just reviewing kind of with a little more detail, I hope. So that is your online learning module for this week. So you should spend 1 hour kind of reviewing those videos and doing a summary. There is a PowerPoint presentation here that I didn't give, but it's just to give you something textual to study from. A lot of what is here, I sort of put into the online module. Okay, so we've already talked about the lipid bilayers here. Some of it we've already covered, right? Ew, gross. All right, so that's your online learning for this week. Okay, so your online learning for next week before your test here is your prokaryotic cells, which is the bacteria. Okay, so there is a review here, a small review. Now, it's nothing major, it's just some basic questions that you should be able to understand. Okay, so these are not the way the questions are going to appear on your test. Okay, your test questions are going to be similar to the practice questions I do in the lecture activities with like the live lectures with me. Okay, but these are just giving you ideas of what the topics are so that you make sure you're studying on the right kind of line. Now, I did not prepare that study sheet. That was prepared by a professor who taught the course before me. I did prepare this study table. Again, my learning style is very visual, so I remember things in kind of locations and comparisons. So I started making this for you to kind of go through it and, cell membrane, here's its description, here's its function, here's the type of plant, animal, or bacteria that it has, that kind of thing. So if you want to fill out this table as a learning practice tool, you can do it. There's no marks for it. You're not handing it in. So you don't need to do that. It's not required, but if you want to use it as a learning tool, feel free to kind of do that. Okay. So when we get into the new learning, because these are all reviews for you, we start out here, comparing prokaryotes and eukaryotes, learning about the different types of bacteria. So when we put them under the microscope, we have to see them with color. So how do we apply the color and why does it show up a certain way? Um, and then how bacteria flagella work and the different shapes of bacteria. So this is really an introduction to the world of bacteria. It's not very detailed, but it's there, and it would be something I could ask test questions from. Okay. So we have one last thing. I want to tell you a little more about the test, and then we'll see how much time we have left. Okay. So your test. Everybody ready? You're still awake? You're listening? Your test will be, it's available now, but do not start the test today. Do not start it just because it's available. Don't click on it until you're ready to do it. Okay. Because once it opens up, it's very difficult for me to give you a second chance on this new system. So the test will be next Thursday, and it'll be open for most of the day, actually. I think the whole day. It's open until midnight. Um, I don't think I have a time to shut it off. I think it's actually open till midnight next Thursday. Okay, so the test will have multiple choice questions similar to what we do in class, true and false questions. It will cover the online learning modules, so the nomenclature, lipids, cell transport, and prokaryotes, those four. It'll cover all of the lectures we've done up until now, so classification of living things, macromolecules, and cells, structure and function, like the organelles that we just finished today. It's a total of 40 questions, 40, and it's going to take about 12 to 13 questions from each section of the course. So there's three main lectures, so I'm going to take 12 questions out of each section. And you won't get the same questions as your friends in the course because I've got like question banks. So I said, here's 25 questions. Choose 10 of them for each student differently and give them 10 questions. Okay, so they might be a little different. And then it'll also, A, B, C, D might show up in one order for you, but if your friend does happen to get the same question, it might not still be A, B, C, D. So you have to sort of be careful if you're trying to be dishonest, I'll say it that way. But you get 50 minutes to do the test. Okay. So this is where, yes, it is an open book test. Yes, you can look at your notes and your lecture slides, and you can go to the online content. You can look it all up, but you will not have time to look up every single answer. Okay. So I suggest you do the test as best you can with the knowledge that you've studied. And then if there's one or two questions that you really don't know, go ahead and look it up. But you have to really manage that time. Okay. So the goal that when we're writing test questions is one question per minute. So if you have 40 questions, I've given you 50 minutes. So it's an extra 10 minutes kind of leeway and grace. I can't leave it open for 2 hours because then you're not going to study at all and you're just going to go and look everything up at the last minute. And then you're not really learning it the way I need you to learn it. Okay. So that would be kind of what would the expectation is. You get one chance and you can kind of do that. And that's all I'm going to do today. I'm not going to start the next section of lecture. We'll leave that for next week. Um, so if there are any questions about the test, the online learning, the course, let me know and I can answer them now, or you can send me emails and I can answer them